Hi students, welcome to week 9 of BIOL-11. We're going to be covering animal evolution. Alright, so in our last video, we talked about the different techniques that scientists use to study evolution in animals. So for the rest of the semester, we're going to be learning about and discussing anatomy across different animal groups. Normally, this would begin our dissection segment of the semester. So some people love dissections and some people hate dissections. So I don't know if you're happy or you're sad that you're missing out on this part. But I'm going to be using different videos of dissections that people have recorded, since I don't actually have access to specimens this semester to do them myself. And there are actually a lot of people who are much better at dissections than I am, so you're in luck. You don't have to deal with the smell, which can be considerable, and you also have expert technicians doing these dissections for you. So biologists trace evolution using these tree-like diagrams shown here, which is also similar to the image that is used in your lab manual. So these trees basically divide organisms based on a trait. So at each branching, these organisms are grouped by that specific trait. So in the first branch here, for example, we see one of the most basic ways that you can categorize anatomic patterns, which is the number of tissue layers. So we have, during our development, we have 3 layers of tissue. Sea sponges actually have no true tissue layers. The cnidarians, which include jellyfish, actually have 2 tissue layers, and then the rest of the tree, like us, have 3 tissue layers during development. And then we can see how closely related these organisms are based on the branching patterns that we create using these trees, just dividing animals by trait. So as we move up that tree, we discuss different traits. In our discussion last week, we discussed studying the embryological development to better understand evolution. This includes tissue layers, like I just mentioned. It also includes body symmetry pattern. So this is whether an organism's body displays a kind of symmetry and what kind of pattern that symmetry takes. So this can either be bilateral, like the beetle shown here. So the beetle can be equally divided into 2 symmetrical parts by dividing down the midline of the body from the head to the tail, or it can be radial, like the coral polyp shown. So this is symmetry based around a central axis. If you think of an axis going through that coral polyp, through the middle, and then if you divide the organism through that central point along that axis, you'll always have a symmetric division. So we also look at body cavity formation during development. So if you look at this diagram, you will see the blastula, which is a stage in development, and it will actually turn in on itself to form a tube. These tubes eventually have openings, and whether these openings, the first opening develops into the mouth or it develops into the anus, is how we divide animals into either the protostomes or the deuterostomes. So we also divide animals based on whether they possess a backbone or not. So invertebrates do not have backbones, and vertebrates do have backbones. For the rest of this lecture, we're going to focus on invertebrate anatomy today, because we're going to talk more about vertebrate anatomy when we begin our mammalian segment for the fetal pig dissections. So we'll just focus on the invertebrates today, excuse me. So this includes the mollusks and the arthropods. So mollusks are a group of invertebrate animals which are defined by their 3-part body plan. So this includes the ventral muscular foot, which is used for locomotion. So if you think about the snail pictured here, you can actually see the foot where they move. It's basically where their body contacts the ground and they sort of just slide along. It also includes the visceral mass. So this contains the internal organs like the stomach, the heart, and then it also includes the mantle. The mantle is a thin tissue that encloses the visceral mass, so you can think of it as like the skin of the slug or the snail. And in some cases, this mantle secretes a hard shell. So this can be seen in clams, but it's also seen in snails. So the mollusk group is also divided into different subgroups. This include the grazing marine herbivores like the chitin shown here, bivalves, which include clams and scallops, gastropods, which include snails and slugs, and then the cephalopods, which include squid and octopus. So I've posted the link to the O-Worm YouTube video channel on Blackboard. I really like the videos from this creator. I think they're going to help you complete everything that you need to do in the lab manual dissection portion, and still give you the experience of what it's like to just sort of cut into something to try and figure it out. I put the direct link to the squid dissection because I wanted to relate it to the octopus video I also posted. But they also have a clam video on there, a crayfish video, and a grasshopper, and they're all really great. So these are all the different structures within a clam. You'll notice that it has similar organs that we do. You can recognize the kidney, the heart, stomach, esophagus, and so through our study of anatomy, we can study how these different structures have changed throughout evolutionary history as you move from simple organisms like the sponge to the clam, all the way up the until you get to the higher order organisms like primates and humans. So we also have the anatomy of the squid. In the videos, they will point out all of the different structures so you can recognize them. That's it for the anatomy of the squid. So I'll move right along since you're going to be watching those other videos as well. I don't really want to make this video too long. The arthropods are categorized by having paired and jointed appendages and a hard exoskeleton made out of the substance known as chitin. So when you think about the arthropods, you can think about crickets, lobsters. These animals have appendages like legs and their claws. They have two of each, so they're paired. And then they also have joints very similar to our joints, which help facilitate movement because they have this hard exoskeleton. If they did not have these joints, it would be pretty hard for them to get around. So this group also includes three subgroups. We have the arachnids. These include the spiders and the scorpions. We also have the myriapods, which include millipedes, centipedes, and the other insects. And then we have crustaceans. So this includes our old friend, the pillbug, crabs, lobsters, and also barnacles. So if you are a fan of eating lobster, the crayfish dissection video is probably going to be pretty familiar to you. I don't actually eat any seafood, but I've definitely used my knowledge from doing crayfish dissections over the years to help my friends and family take apart lobsters. I don't think you'll really ever look at it the same way after you dissect the crayfish, and really get a look at all of the structures inside of that body that you usually throw away. So check out the crayfish dissection video. You can also check out a grasshopper dissection video. We usually don't get to dissect our grasshoppers because we just have the one specimen that's preserved, and we usually just check out the external structures. So one of the important structures I want you to pay attention to here in grasshoppers are the spiracles. So grasshoppers and crayfish are actually very similar because they are both in the arthropod family, but they're adapted to two completely different habitats. So the crayfish lives underwater, And as such, they have gills to facilitate gas exchange, which help them take up oxygen from the water. And grasshoppers obviously live on land. So they've developed these spiracles. So these structures are what this animal uses to facilitate gas exchange. They're basically holes to the external environment. Air can flow directly into the muscles of the legs and the wing. So these are the most active parts of the grasshopper. So this is where the oxygen is going to be most desperately needed. So it's pretty neat. Rather than having gills to facilitate this, they just suck in the air straight from the environment. So a very interesting process that takes place in biology is the process of metamorphosis. So this is most famously seen in butterflies. We all know the progression of the different forms of the butterfly life cycle, where it's the caterpillar larvae, becomes the pupa, and then emerges into this spectacular butterfly. So this is called a complete metamorphosis. So insects that undergo complete metamorphosis undergo a very dramatic change in the appearance of their body. So the juvenile organism does not resemble the adult version at all. The caterpillar larvae just doesn't look anything like the butterfly. Other insects also undergo a different process of metamorphosis called incomplete metamorphosis. So these organisms don't make a complete change in appearance. The juvenile form of the grasshopper very much resembles just a small adult grasshopper. These organisms will undergo molt throughout their life to increase in size because they need to get rid of their exoskeleton in order to grow bigger. But they still look the same. There's not this dramatic change in their appearance. So the final group of animals for today's lecture is the chordates. So these organisms possess 4 characteristics that they all have in common. This includes the presence of a dorsal tubular nerve cord. They also have a dorsal supporting rod called a notochord. So this can be present at any point during development. Sometimes it goes away. All chordates have a postanal tail at some point during development, and pharyngeal pouches during development. So these structures actually become the gills in the animals that have gills. So this group includes the tunicates, which are actually a marine invertebrate animal that includes something called a sea squirt. And there's also the lanceolets, which are fish-like animals. And then it includes the vertebrates, which include the lampreys, cartilaginous fishes like sharks, ray-finned fishes, which include things like tuna, lobe-finned fishes, which include the very bizarre lungfish, amphibians, reptiles, and mammals. So not all chordates are vertebrates. The vertebrate is specifically an animal that has the spinal cord, which is surrounded by either bones or cartilage. This term comes from the name of the bones that make up our backbones. They're called the vertebrae. All right, so the activities for this week. View the dissection video that I posted on squid anatomy, and click around on that YouTube page to check out the videos for clam, crayfish, and the grasshopper dissections. Then you can use these videos to complete all the questions in your lab manual, just as if you were dissecting these specimens. So if you have any questions, let me know. I'm available by email, and of course my office hours Friday from 1 to 2. Have a great weekend.